We're going to look at protein synthesis part 2. This is page 7B and 8B in your notes. All right, the important concepts from the previous units, it just tells us that amino acids are the building blocks of protein, of the macromolecule proteins, and the amino acids get linked together by peptide bonds. All right, so all a peptide bond means is that you have two amino acids linked together, and this happens by dehydration synthesis. So you can see the hydroxide and the hydrogen gets pulled out to form water, and then the peptide bond is created between the two of those. Okay, so it goes on to talk about that proteins have to be folded in order to work, and if you'll remember that, we created those green fluorescent proteins. And remember, we had to fold those all up. You have the primary structure in a protein just means that you have an amino acid linked to an amino acid linked to an amino acid, and they're very straight, okay? So it's just in like a single file line. Then your secondary structure is where you form hydrogen bonds between parts of that are very close together. And so it looks like it refers here to a pleated sheet. So it just makes it have a folded appearance. And then after that, you have a tertiary structure, which is where you have what are called disulfide bridges. And they just form in between some of those folds. They become attracted to each other, and then they fold there. So in getting into the notes to translation, remember that we said translation, and just thinking about that in layman's terms, that is taking one language, converting it to another language that is understandable for you. So translation in this case takes the language of the codon, and it's going to translate that code that's on the messenger RNA. It's going to translate it into the name of an amino acid. Okay, so the way that happens is going to happen on the ribosome. The messenger RNA is going to leave the nucleus. It's going to go out to the ribosome, and it's going to take on that primary structure form. So it's going to be a single file line of amino acids that are needed for making that protein. All right, so your messenger RNA is here, and it links onto the ribosome. And then you have transfer RNA, tRNA here, that are going to have amino acids attached to them. They are going to come and bring the amino acids to the ribosome based on whatever the codon is down here. They're going to have a matching or a complementary anticodon that matches up or pairs up with the codon on the messenger RNA, and that way they know that it's kind of like a puzzle piece. If those fit together, they know that they have brought the correct amino acid. Okay, and the place that those amino acids come from is just like things that we eat, our digestive system. They break those down by catabolism. Remember, catabolism is like catastrophe, breaking stuff down. And then anabolism is going to be where we build those things back up, okay? All right, so there are 61 possible codon combinations. One thing that your notes mentions is that you can have something called inosine, and what it means is that it acts kind of like a wild card. And it will be on that in a codon, that third lettered sequence right there, okay, like you have UAG or something, okay, that third letter can be inosine, and it is able to pair up with some different things. It won't pair perfectly, so it's almost like forcing a puzzle piece into a place that's sort of close but not really exactly right. And for that reason, they call it a wobble effect. But that does happen occasionally, but that's not, I guess what we would consider the norm, the typical rule. Okay, so the typical rule is going to go by those base pairings like we've been learning about. All right, so this is a transfer RNA. You can see that it's got a 5' end and it wraps around to the 3' end. And then if you look here, this is a transfer RNA, another picture there showing that it's folded. And then you can see down here that you have your anticodon there. And this is where the amino acid would link to right there. Okay? All right, then you have, here's your codon chart again. These are the codes for all of the different amino acids. All right, so you've got your messenger RNA that's going to leave the nucleus. It goes to the ribosome. And this is showing you here how that the anticodon is going to match the codon. So the codon there is in pink, AUG. The anticodon on the bottom of the transfer RNA would just match up with that, pair up with that. All right, we're going to look at the ribosome structure, and this is something that we drew in class, okay? It's made up of a small subunit, which is here. The large subunit is here, and it has 3 different sites. You have an E site, the P site is going to be there, and then the A site. So E, P, and A. The A is going to be where the next transfer RNA is added. Okay, so if we look again at this picture here, this will kind of help you understand how this RNA is going to help in elongating, okay, making longer this protein, all right? So you have a transfer RNA that's going to come in, okay? And the protein's going to be made on the P site. So P for protein. This is where we're making the protein. You can see there the link of the amino acids. The transfer RNAs, when they come in to the ribosome and match up their anticodon to their codon, they're going to attach to the A site. That's where the new transfer RNA that's brought in is added. So you can see that here. This one's going to be added. It recognizes the codon. And then it is going to continue to shift to the left. So you have the transfer RNA that was added that will then shift to the P site, okay? And then they're going to be transferred then they continue shifting to the E site where they'll exit. So in the meantime, what's going to be happening, you'll have amino acids, see right here, that are linking up together. So it's almost like you have a chain of amino acids and you're passing the baton to the new transfer RNA. Saying, okay, here's the link I've already made, the chain I've already made, add yours to it, okay? And then it's going to go on to the exit site, E for exit, and get out of the way. And then that transfer RNA is going to move to the P site. Then a new transfer RNA is going to come into the A site. It's going to be adding an amino acid. It's going to, again, hand off this chain of amino acids that have been building to the new guy that's just come on board, okay? And then it's going to continue to go on down the E site, and it's going to exit, and the new guy then takes the place where the protein was. So in this way, you just keep adding amino acids and adding amino acids to make them longer each time, okay? So what we've had happen, we've had initiation, which is where that small subunit on the ribosome attaches to that 5' cap. Remember, it's like the hard hat that we put on the end of the messenger RNA. It's going to attach to it, and then it's going to start all of this process. You can have something called GTP that's going to provide the energy for that. All right? That's guanosine triphosphate. It acts very much like ATP. They're both energy molecules. Okay, so you'll see that there for initiation. And then elongation is what we just looked at, where we just keep adding amino acids. We have the E site and the P site and the A site. And then termination is going to be where we terminate the process. We have made our protein. It is long enough. Amino acids have been added. We're done. And so we want to stop making the protein. So what's going to happen there is you're going to have something called a release factor that is going to come in and add itself to that new site. So instead of having a new transfer RNA come in, you have a release factor come in. And so notice that it does not have an amino acid added to the end, okay? So it just tells the protein, hey, stop making. Okay, so the release factor just signals for it to terminate. Now, you can have something called polyribosomes, and just from your prefix, we know poly means many. So if you look here, it's got lots of ribosomes working on the same messenger RNA. That's if you need the same protein and you just need lots of them. You'll have several ribosomes working on that same messenger RNA, getting those codes, making that same protein, just lots of copies all at one time. So it gets lots of work done at one time. So I want you to look at this real quick just to kind of recap where we've been. The DNA template is what we start out with, okay? And that is just all of our codes that you go in and by transcription, the messenger RNA, remember by transcription, it's going to come in and copy that code from the nucleus, okay? And it's going to then carry that to the ribosome. So it comes in and remember it works 5' to 3'. Okay, so you've got the T going with the A going with U, C with G, okay, and you just keep going and paring those down. All right, so then we read the messenger RNA. The messenger RNA is what we translate. So that is translation, and it occurs in the ribosome. All right, so AUG on your codon chart translates to methionine, which is also the start codon. So AUG will trigger and signal the protein, hey, start making. Then you have GUC, which will signal for valine. And remember, the way that it knows which one to bring is because the transfer RNA is going to have these anticodons on the end. And see, you've got AUG, well, UAC is going to fit right with that, just like a perfect puzzle piece. Now, the question that it says down here, it asks you, does the DNA sequence here, the $1 million blueprint, look like the anticodon sequence? Well, it makes sense that here's the DNA. Remember, we had to pair it with a complementary strand, and then you pair that with a complementary strand down here for the anticodon. So it really is going to mimic what you had to begin with. So that's just kind of its way of checking and making sure that it was similar to the original instructions. Except, of course, we're going to have U's on the RNA rather than T's like the DNA. Okay, the last thing is the post-translation modification. So this is how we have to modify the protein to make sure that it's able to go out into the world and do its job like it needs to, okay? So one of two things can happen. Either it can enter a chaperonin, which if you think of the word chaperone, a chaperone is somebody that makes you behave and do what you're supposed to do, okay? You'll have a polypeptide, which means that it's a protein, that's going to actually go down into this chaperonin, which is like a cylinder, and it makes sure that it gets folded up like it's supposed to. If it uses this, it's going to be a protein that stays inside the cell. It is not being made to leave the cell. Now, it can also be taken to the rough endoplasmic reticulum. And remember, the rough endoplasmic reticulum is made for transport of proteins. Remember that? Okay, it transports proteins through this folded system of membranes. So if that's going to be its destination, if its destination is to leave the cell, then there will be something called a signal peptide. And basically, it is like a siren that signals and yells out, hey, take me to the rough endoplasmic reticulum, okay? And so it will go there. This signal recognition particle will pick up on that, and it will guide it, you can see, and it takes it to the rough ER, okay? And so then it will be put into a compartment and fixed so that it can leave the cell and do its job outside the cell. Okay, so big picture, this is the picture that I gave you in class. And you can see that it goes through transcription that takes place in the nucleus with the DNA. It goes through where we modify that RNA. We get the 5' cap. We get the poly A tail. We remove the exons, the introns. We splice those with the spliceosomes, and then all of those exons are going to be put back together. They're going to leave, go to the ribosome, where transfer RNA is going to come and bring those amino acids, and will through translation, and we'll get our protein with all those amino acids added together.